I'd like to go back, first off, to thank Ciami for the invitation. This is my third year here in Riccione. It's always a huge pleasure to be here and talk to all of you and answer your questions on gene therapy. And then I'd like to also thank all of those who are following us now via the internet. For those of you who don't know me, I'm Federico Mingozzi. I work in Paris, France. Officially, I'm an employee of the French government, but I work at the Geneton Institute, which is a non-profit institution established by the French Association Against Muscular Dystrophy, which is involved in the development of therapies in general, most especially gene therapies for rare diseases. So today I'm going to talk to you about a program that was conceived a long time ago, several years ago. We've been talking about gene therapy for CN for several years, and I moved from the US to France 3 years ago, and since then I've been involved in this. Now, this is what I'm going to talk to you today. First, a short introduction on gene therapy, and then I'll delve into the details with respect to the gene therapy program for Crigler-Najjar. Now, without being too fussy, bear in mind that the idea of gene therapy arose from the knowledge of the human genome. As we've started to understand more about genes and the genetic contents of the nucleus of the cell and its implication in diseases, hence we've started to think of how to solve genetic diseases through the so-called gene therapy. In the case of Crigler-Najjar, we've already heard a beautiful introduction from Lorenzo. The gene responsible for the disease is UGT1A1, and the goal of the gene therapy is to restore the operation of this gene in the liver, because this is where this enzyme is synthesized, and that's why liver transplantation works in this disease. So, to do that, to transfer a gene, you can use several things. Today, the most effective vector to transfer a gene in a cell is a virus, because viruses have evolved as perfect or almost perfect machinery to infect a cell and transfer their genetic information in the nucleus of the cell. So what we've done is take a virus and engineer it to transport the UGT1A1 gene and take the virus into the liver. So, having said that, the viral vector we're talking about is AAV, that's its name. It's an adeno-associated virus, and it derives from a virus that is present in nature. It occurs in nature. So it's an isolated virus. We've engineered it. So there's the virus, the therapeutic vector, which is produced to be injected in the blood, in a peripheral vein, and reach the liver to correct the disease. What happens, and well, quite obviously, this is a sketch, there's nothing real, but just to give you an idea, the virus enters the diseased cell and transferred the corrective gene, UGT1A1, into the nucleus. As I said earlier on, the viral vector, AAV, is a vector that derives from a virus which occurs naturally, and today this is the most effective gene therapy vector to transfer genes directly into an organism. This vector comes from a virus which does not independently replicate. So it's a virus which is not pathogenic and is not hazardous, because normally we are infected, for example, by this virus without realizing that we are so infected. Importantly, we can pick these AAV vectors and produce them in a sufficient amount to do gene therapy. In this test tube, you can see these two lines, and these, most especially the one at the bottom, are the vectors used for gene therapy. The use of AAV vectors in the clinical scenario, i.e. to treat genetic diseases in man, is not new. There have been several studies for very many years. I'm talking about 20 years or more that we've been talking about gene therapy. And so many studies in preclinical models. But now we also have many examples of clinical studies in which AAV vectors are used, like in Duchenne, muscular dystrophy, Pompe disease, hemophilia, and hopefully very soon Klinger-Najain, brain diseases, retina diseases, the liver disease is going to be one of the upcoming gene therapies to become a drug, and so on and so forth. Currently in Europe, there is a gene therapy that has been approved as being a drug for the treatment of lipoprotein lipase deficit. Now then, quite obviously, as is clear for all, I think, the idea here is to have a therapy directly for the liver where the UGT1A1 is synthesized, and also because the liver is one of the places where most of the plasmatic proteins are synthesized. So it's a small, very active factory that produces proteins. And it's also the place of the synthesis of the UGT1A1 enzyme. Now we know that we can reach the liver cell and the liver with these AAV vectors quite simply through an injection through a peripheral vein. It's like an intravenous injection of any other protein. And then we've also have data in man saying that the transfer, i.e. the gene therapy directly to the liver, is safe and can be effective. For the time being, what do we know about gene therapy in the liver for man? Well, most of the knowledge we have relates to the studies for the treatment of hemophilia A and B types. With respect to this, the liver is the place of synthesis of the coagulation factors, and so the researchers have used adeno-associated vectors, AAV, to transfer the genes of these coagulation factors directly to the liver. And right now we have at least 5 clinical trials underway in which AAVs are being used to transfer one gene to the liver. So we have plenty of data partly published or being produced. Now, I will briefly summarize what we've learned up until now with respect to the clinical context of gene therapy based upon clinical trials. We know that it is possible to use an AAV. Number 8 is a specific type of this AAV. We call it serotype. It's the one that will be used in the Klinger-Najain clinical trial. So we can use these vectors intravenously injected to reach the liver. We know it's possible. The data we have up until now shows that if we reach the liver in an efficient manner and correct a disease like hemophilia and have a stable level, this will last at least 6 years. I say 6 years because we don't have longer follow-up. So We've had hemophilic patients for 6 years treated with liver-directed gene therapy. We also have data in dogs suggesting that the effect is lower than 10 years. But obviously, like in the case of liver transplantation, in some cases, it's a book we are writing now. We don't have very long-term data because we started 10 years ago. Like with all therapies, there always are safety considerations with respect to side effects. And gene therapy, how shall I think of it? It's like a transplantation. I mean, we all know that when we're infected by a virus, our body reacts against the virus. And so, in the case of AAVs, in some patients who've received the vector, there has been an increase in the liver enzymes. I mean, a slight, very slight, and not associated to any symptoms, a very slight liver inflammation that has been treated with the administration of cortisone for 4 weeks. And the inflammation has gone away. So we know that with respect to gene therapies with AAVs, in some cases we have to use cortisone for 4 weeks. So, this information deriving from clinical trials, I mean, all of this is very important for what we want to do here today. First of all, today we know that we can reach the liver in man and can transfer a gene in a stable manner for several years using this technology. We didn't know this a decade ago. We also know that in order to correct the Crigler-Najjar disease, we don't have to take the UGT1A1 enzyme levels to 100% like a healthy person. We can simply have it like above 50% of its activity, and we know that this is doable. We can hit this goal with gene therapy. Because with other diseases, we've been able to achieve levels of at least 5%, not 50%, 5% of the normal activity. And it's also important to know that today we know better the possible side effects of gene therapy, like the problem of the immune response against the virus. And this was treated with the administration of corticosteroids and cortisone. An important thing to say with respect to this slide is that one of the benefits of gene therapy is that it does not interfere with the treatment of the Crigler-Najjar syndrome. It doesn't interfere with phototherapy, and in case of failure of gene therapy, quite obviously, transplantation is still an option. Let us now dig down into the details of the gene therapy project coordinated by Geneton and co-workers. I'm going to show you some data, not a lot, on animal models, and then I'll move on to make some comments on the progress of the project. So, again, we've used an AAV type 8, which includes the UGT1A1 gene, and the idea is to use this vector to transfer the gene to the liver. In point of fact, we have verified the effectiveness of this therapy in multiple animal models, but one of the models that we have used to test our technology is the gun rat. Probably many of you know it. It has a spontaneous mutation in the UGT1A1 gene, so it has Crigler-Najjar syndrome, but we've also used a rat model that was developed by Andres Muro at Trieste. And we've collaborated for a long time on this project. What we have observed is that after a single administration of our vector, there has been a lowering of the levels of total bilirubin in serum, and this lasted. Quite obviously, we've verified growing doses of the vector, but at a dose higher than this number, which doesn't mean anything except for the fact that the dose is similar to the one used in man. Anyway, we have observed a lowering of the levels of total bilirubin in the serum, and this has lasted, as I was saying, up to 1 year. We have a 1-year follow-up with the animals. It's a prolonged lowering of the levels of bilirubin over time. Up until now, as you can see, well, I haven't shown you that much, but where are we now? Well, we've completed the effectiveness study in rats. It's a 1-year study. We are conducting studies in animals in the neonatal period because the rats that we've studied up until now were equivalent to young adults, humans. Quite obviously, we've studied the interaction of the vector with cortisone in the event of it being necessary to use cortisone in the clinical trial. And we are conducting repeated dosage studies, which is simply something that is required by the supervisory authorities. They need to verify whether or not we are capable of doing gene therapy in the second time in the event of there being a decrease in the effectiveness. And finally, I'm going to show you the results of the toxicity studies. This is required by the law, and in practice, the drug, the one that we want to develop for the Crigler-Najjar syndrome, is administered at doses equivalent to the highest dose that will be administered in the clinical trial, or at doses which are 5 times higher. In this case, we've taken rats, and we've administered to them the UGT1A1 vector at the maximum dose provided for in the clinical trial, or 5 times higher. And then we've had a control group to which we've simply given the saline solution in which the vector has been diluted. And thereafter, we have examined the levels of toxicity at 7 days. This is the acute toxicity, then the chronic one at 3 months. And then we've also had a group of animals followed up for 6 months. As I said, all of these studies are required by the supervisory authorities, and they are used to evaluate toxicity, the immune responses against the vector, as well as how this vector is distributed throughout the body, in the various organs of the body. And again, how this vector over time is dispersed by the recipient. So we, for example, measure the presence of the vector in the urine and for how long it stays there. So far, so good, because no acute or chronic toxicity has been seen. So we haven't seen any changes, significant changes in terms of liver enzymes in the blood of the animals. We are considering the biodistribution of the vector, but we already know it. The vector, once it is systematically injected, it ends up in the liver and everywhere. We know it. And in practice, this study will come to an end in September this year. One of the most difficult things, and I'm saying so based upon my experience, one of the most difficult things in doing a clinical trial of gene therapy is having a product for humans. So the production of clinical quality vectors has been one of the most important factors in this project. There's a mistake on the slide. These are not the right cells, but what is done to do these recombinant vectors is grow them in bioreactors, large vessels, 100 liters vessels, and then we add there too the reactants that we need in order to create the virus, which is then purified. Well, it doesn't tell you so much, except for the fact that now we have a process to make vectors. It works. We've tested it several times. We prepared several batches of vectors. It's not a clinical vector, but it's an in-development vector. Now we are ready to transfer the production process to the team of persons who will produce the drug. So the production process of the clinical batch, as I said, is the following. So we have a 200-liter scale process, which is not that little. This is the result of the work of Matthias Hebner at Geneton. Great work. We've ordered the raw materials to make the vector, and they're expensive. 800,000 euros of raw materials that will be delivered in July this year. And now we are transferring the process from R&D to production proper. And in practice, production should end at the end of this year or beginning next year. We're talking about large-scale productions, some million euros, including for a rare disease like Crigler-Najjar. So some million euros to produce the vector. And then I'll give you some more details on what's next. Many of you probably know that we've organized across Europe an observational study, and I'm going to talk to you in a moment, and then I'll be talking to you about what will be the future clinical protocol. These observational studies completely disconnected from the clinical protocol. Now then, I'd like to thank the participants in the CureCN network, as we've called it. It's a network of European co-workers and clinicians. In France, we have Geneton for R&D, and then Professor Labrune in Paris as clinician, and the Patients Association, quite obviously, in France. In the Netherlands, we have Peter Bosma and Dr. Boyer, Amsterdam, as clinician, and the Patients Association. In Italy, we have as researcher Andres Moro, and as clinical centers we have Bergamo with Lorenzo D'Antiga, and Naples with Nicola Brunetti Pierri, and the Patients Association, once again, that has helped us so much in this project. And finally, in Germany, we have a clinical center in Hanover with Mr. Ott and Dr. Baumann. I'd like to start from the clinical protocol, and then I'll move on to the observational study. So the future clinical protocol for gene therapy will be a phase 1 to open label study. It means that it's a study in which for the first time this therapy will be administered to humans. Open label means that there will be no placebo. All will receive the active ingredient, the drug. It consists in the intravenous injection of the vector that takes the UGT1A1 gene. The number of patients is about 17. The main goal, since it's the first time that this vector is used for humans, so the main goal is safety. The secondary goal is effectiveness. It's a dose escalation dose. We will have a starting dose, and then the doses will increase over time. But every patient will receive the vector only once. So growing doses of vectors will be administered starting from a dose 2 multiplied by 10 to the 12th Vg/kg. It's a dose that has already been used in man. We already know the safety and effectiveness profile with respect to hemophiliacs. And in rats, i.e. in animal models, it's turned out to be effective in the long term. So we don't start from a dose that is not therapeutic based upon preclinical models, because this is what we have agreed also with the authorities, regulatory authorities, with the European agency. It would not be ethical to use a non-therapeutic dose for an ultra-rare disease like this one. Quite obviously, one thing to be borne in mind, an important one, is that it is the first time that this vector is being administered to humans, so we cannot guarantee anything. We can only start from a point in which we believe that there can be therapeutic effectiveness, but there is no guarantee as to the effectiveness. This slide is the chart of the clinical trial, and what matters is that it will be necessary for patients to sign an informed consent, but the drug, the vector, will not be administered immediately after the signature of the informed consent. There will be an observation period that will be used to collect data to evaluate the effectiveness of the therapy later on. So every person included in the clinical trial will be his or her own control for the purposes of measuring the effectiveness. So when we talk about clinical trials, we always talk about inclusion or exclusion criteria. Quite obviously, the molecular diagnosis is important. So the persons who will be enrolled in the clinical trial will have to have a mutation in the UGT1A1 gene, will have to be under phototherapy, and the age will be 10 years or more, because once again, this is a trial in which the drug is administered to humans for the first time. And the very first patients included will be of age. And then we will also include younger patients once the approach has been demonstrated to be safe and effective. As I said, the patients who decide to participate in the clinical trial will have to agree to use contraceptive methods in the 6 months following after the administration of the vector. This is simply a safety or precaution measure. And finally, they will have to sign the informed consent. I mean, the doctor will explain all of the risks connected to gene therapy, and thereafter they can think of it very well, and if they decide to be enrolled, they will have to sign the so-called informed consent.
In terms of exclusion criteria, well, we have fibrosis, HIV, chronic diseases, heart diseases, and also in case of severe disease, right before the clinical trial, it is better to wait some time. And also, we consider that if a person has already been enrolled in a clinical trial, he has to wait at least 12 months before starting a new clinical trial. And of course, another exclusion criteria is that a person is not capable of complying with the study, so all the controls and all the exams to be performed. Now I move on to the observational study, because in terms of organization of the clinical trial, of course, we have to collect data, molecular data about the UGT1A1 gene. In the countries who joined the network, and also the neutralizing antibodies for the vector. And I will give you more details about that. So the observational study started in November 2014. The main goals of the study were, first of all, collecting clinical data about the health of the people suffering from Crigler-Najjar syndrome, and also assessing the antibodies against the vector. We have 5 clinical centers: Italy, Netherlands, France, Germany. This is an observational study, so it is in no way associated with the gene therapy study. An additional informed consent will be asked for when it comes to the gene therapy study. So the study was really successful. And I have to say that all of you, and also the coordinator of the Patients Association, participated. So 45 people participated in this study. The results about antibodies were disseminated, but of course I don't have data about that because I do not know the identity of the donors. So what is the problem associated with the antibodies of the vector? Well, the problem is that the AAV virus was first isolated in nature, so we were all exposed to that virus, not exactly to the type 8, which is a serotype found in monkeys. But if we look at the type 2 virus, I think that 70% of us has antibodies against it. So when it comes to the type 8, about 15 to 20% of the population has antibodies. So if they bind to the virus, they prevent it from reaching the liver, so it becomes ineffective. The first study, which will start soon, will not include people having the SAD antibodies for the serotype 8, AAV8. However, in the future, we have procedures like plasmapheresis that can be used for removing the antibodies temporarily, so that the gene therapy can also be applied to these patients. However, this will be later studies, so this is not done in the first phase of the experimentation, but only later on. When it comes to the next steps, so this is where we are today. The green in the slide represents the observational study, so the non-clinical study. So we have the study on rats, which has been completed. We are now assessing and evaluating all the samples. Then the process development concerns the production and the definition of the ideal procedure for the vector. And in the months to come, so we will have to start the production of the clinical batches of the vector. And as soon as they are ready, we can start with, and we can actually submit the application for the clinical trial that will start next year. In terms of the results obtained so far, we have obtained the orphan drug designation by the European community, the European Union. So we have been recognized as an institution, as a body working on a rare disease. So for the time being, this does not make any difference. But if this therapy is going to become a drug, then of course this designation will be very important. We have already discussed the clinical protocol with the European Agency of Drugs, and we received indications on how to change our trial to make it even more effective in terms of what we can learn from it. We have defined the production process. We are ordering the raw material. The toxicology study will be completed soon. We are also carrying out other studies on newborn animals. The observational study is about to be completed. The 5 centers, 2 in Italy, have been identified, and the clinical protocol have been discussed and with all the physicians who collaborated through the network. What about the future? So the future steps. Well, first of all, we would like to contact all the drug agencies in France, Italy, in the Netherlands and Germany, so that we can go ahead with the clinical study, because in Europe we do not have central approval, so by the central agency, but every single country has to approve it for the clinical trial to start. We are also considering all details. So, for example, we are contacting all the pharmacies in hospitals in order to define the preparation of the drug, and also in order to define the authorizations needed in order to start the clinical trial. Then the production process will be transferred in order to start it, and then this is of course the most important step. But then of course we will have the documents, and the documents will be sent to the health bodies in the member states. So this is all. I would like to thank all the people who have been working with me at Geneton, so my lab, but also clinical development, production, so all the people, the whole organization of Geneton and AFM. So Thierry Bordet is the project manager of the project. So he helped me so much. He's really passionate and dedicated to this project. I also would like to thank warmly the Patients Association. So thank you so much, but also thank to the clinical centers who participated in the observational study, but also contributed to the definition of the clinical protocol. And last but not least, the researchers, who, like me, have been contributed to the development of this gene therapy product. Thank you. All right, so now time for questions. Let's start with a very simple question. So next year, when our meeting will be held, what are you going to tell us? That we are ready, that you have already started? So let's, April next year, if everything is okay, we will be about to start. Honestly, I would say that if we have started, we will be in the observational period. So maybe some patients have already or will have been already enrolled, but I mean, no injection yet. So we will be very, close to it. Okay, so now we have a question by Beatrice. So you mentioned the toxicity studies, and you said that the study was carried out with a minimum dose and then maximum and then 5 times the maximum dose. Yes, we start from the highest dose to be administered in the clinical study. So we started with the highest and then we reach 5 times the dose. So we would have a group with the highest dose and the group with 5 times the dose. And you also said that after 7 days, 1 month, I mean, even with the 5 times the dose, there was no toxicity. Well, yes, for the time being, we have assessed the clinical chemistry, so enzymes, and we also measured, for example, the weight of the internal organs, and no deviations were found. What is missing now is the histology. So at microscopical level, we are going to look at any changes in the organs, and in the same time, we look at biodistribution. But I mean, the vector will be in the liver mostly, but we know that. So I think this is a very good datum. I mean, if we think 5 times the dose, no toxicity, well, I mean, if this was not the case, we wouldn't have continued because, however, this is the maximum threshold. So if we have toxicities, a very high dose, then of course you have to lower the dose because you have to lower toxicity. But do you think we will reach the maximum dose in the trial? Well, the proposal under discussion is that we should start with theoretical dose, but using the dose escalation, so increasing it. And then we see what happens. Well, the best approach ever is considering many different doses so that we can choose the one which is most effective over time. So I would say that the best approach would be to use the dose escalation up to the ones proposed. We also have a question by physician Michele Colledan. I think it is really fascinating what you said. And I'm thinking about the aftermath, so what is going to happen, because when it comes to drug economy, when a new drug is released, so with a new technology, an expensive one, then, the problem is that it arrives after so many other drugs, so regardless of the ones used before. So, for example, we also have a few cases of drugs that, even though they are more effective, they came later, so they are not used in the national health system. So, by definition, and considering the type of syndrome, of course, this therapy will not benefit from scale economies. So do you have an idea about the cost, about the price of the treatment? Well, there are numberless estimations, and the U.S. Society of Gene Therapy is now organizing a number of workshops focusing on the price of the therapy. For the Crigler-Najjar syndrome, the disease is so rare that I think it will not affect the budget of a country. However, a number of proposals have been put forward, and one of them is to make the price of the drug a sort of a fee, an annual fee. So basically paying an annual fee to the developers until it is effective, or some proposed an annual cost. You can use, for example, the cost of the liver transplantation as a benchmark. But personally, I think that no agreement has been reached yet. For some diseases, such as the PASI deficiency, the price was proposed, but I think it was accepted in Germany only because it was very high, and also the effectiveness was not 100% clear. However, the debate is there. The products are very expensive because, first of all, the production process is expensive. Secondly, these are rare diseases, so of course the gene therapy won't make anybody rich, when it comes to Crigler-Najjar syndrome. So this is the reason why we need an association, starting with the first investment, and then maybe a pharmaceutical company will take it. And this model has been used by Teleton, for example, when it comes to primary immunodeficiency. So the therapy is developed by the San Raffaele Hospital, so now are becoming proper drugs at the moment. Any other questions? I have a curiosity. The virus, so the vector we mentioned, carries the gene to the specific chromosome or to another place, and then the information will be read. Well, the vectors are not integrative, so what happens is that the gene remains in the nucleus in the episoma form, so it's like a small chromosome that remains there. So in case of cell division, it is transferred to the cells, but it does not integrate into the genome. So any chromosome can intercept it, but no, because it is not part of the chromosome, it remains far away from it, so detached from it. So you mentioned 10 years in dogs, but this means 60 years for human beings. Well, I prefer to look at data concerning human beings, and for the time being we have 6 years, which is a good result already. We have another question by the Venturi family. Well, I want you to repeat it once again. The dose 2 by 10 at 12. So this is the safe dose, and this is the first to be injected, right? But this would stimulate the 5% of the enzyme that we need. Well, I cannot tell you this. I can only tell you about the data I have already seen. So I cannot guarantee the effectiveness in the clinical study. Because, 5% is not that much. Well, we have seen that in other diseases. We have seen the total correction in rats, but of course rats not humans. So I can tell you that the dose did not produce significant side effects in human beings. And I can tell you that based upon the preclinical models and what we have already seen, there is likelihood to be effective, but I cannot tell you that it will be effective. Because, this is the purpose of the clinical trial. The clinical trial will help us to see whether what we have seen in rats apply also to human beings. We have a question from Katia. Well, I go back to the money issue. Since the disease is so rare and given the very high costs, do you think pharmaceutical companies may stop the production of the drug because they have no return on the investment? Well, no. Why would they do that? Today, they are very interested in rare diseases. So once we have demonstrated the effectiveness and of the project, there's no reason why pharmaceutical companies may stop such a gene therapy project. So I would say no. So to your question. Regardless of the effectiveness, what are the real risks? Do you have any ideas about risks? The risks we know are that using the dose we mentioned in human beings, we have seen that it was necessary to administer cortisone 7 weeks after administering the vector, because the hepatic enzymes increased. However, it was a minor asymptomatic increase, so we found out only through the lab tests. At the moment, we don't know if there are other side effects. A possible one is mutagenesis. So the vector may enter the genome, and this may happen, and this may produce side effects. Such as the tumors, but this is a theoretical risk only because we haven't seen it in dogs or monkeys, and not even after some years, and we haven't observed it in humans either, but these are the potential risks nonetheless. We have another question. The 17 patients will be administered the same dose? No, we will split them in groups. So the idea is to have groups of 4 patients. So the first 4 patients will receive the starting dose. Then we will have an observation period. Then we will start off with a higher dose. And then we will have another group of 4 patients. And this group will start after a few weeks. In between the doses, we will have an independent assessment committee which will be evaluate the data. And so before going ahead with the next dose, we have to wait for the authorization by this committee. You were also asked about re-administering studies. Well, this is kind of a standard procedure asked for. So same vector, same serotype, same dose, yes. Well, the regulatory bodies ask for a program in which, for example, you change or you use a different serotype or you block the antibodies response. But however, it is a standard procedure. Okay. Oh, there is another. No, it's time for a gift, but there is also a very last question. I would like to know the dose you mentioned. So although it's not going to be very effective, but I mean, at least a minimum effectiveness is guaranteed. I cannot say yes, I'm sorry, but I cannot tell you yes. I cannot promise you that it will be effective, but you expect it, right? Well, of course, also when it comes to the discussions with the European Drug Agency, well, during the discussions, you know, we said, okay, you have to start with a dose having, you know, some effectiveness. You hope some effectiveness, but of course, you know, when it comes to a higher dose, then safety becomes an issue because it was never tested in humans. So we start with a dose which could work, but I cannot tell you yes. I cannot tell you that is going to be the case, although preclinical studies are good. However, it is not dangerous dose, so it looks like there are no risks for health. And however, if it doesn't work, as I said, phototherapy is not altered, so you can do that. And it is also possible to undergo liver transplantation, so nothing changes.
I understand. As you want.
Quindi se è efficace con la prima dose, volete comunque provare con la dose successiva o no?
La prima dose comunque, no, perché l'ho detto, comunque proverete con una dose maggiore o continuate con quella dose che ha funzionato col primo gruppo?
So in this case, we have considered this possibility, and there are two main hypotheses. So the first is increasing the number of patients to whom we would administer the first dose, which proved to be effective. Or the second hypothesis is to go ahead with a higher dose and looking at the effectiveness and safety for the higher dose. Both approaches are valid, because in some studies, well, they used both. So in some studies, they found an effective dose, for example, for B hemophilia. So they proved one specific dose to be effective, and so they moved on to the higher dose, but then they went back to the first dose. We have not decided yet for our trial, so no final decision yet. But when it comes to effectiveness, what does it mean that bilirubin from 20 goes down to 2 or from 20 to 15? I mean, this is another point of discussion, a crucial one. So in terms of, when it comes to the European Drugs Agency, their approach is really pragmatic. So they said you have to look for anything which is capable of lowering the bilirubin level. But we wanted to focus on the possibility to stop or reduce phototherapy. And this is not always associated, and this at least is what we are discussing about. So the possibility to stop phototherapy is the most important thing in terms of life quality. So we propose to use this as endpoint, so in terms of effectiveness.